Hello, guys. How are you all doing? Good morning. Good morning, everyone. Good morning, Aditi. Good morning, Smart Buzz. Good morning, everyone. Good morning, Anoop. So, how are you all doing? How was your Saturday, Sunday? Good morning, Komal. So, 26 people watching. Um, so, today I've come up with Unit 6, where we'll be discussing majorly about photosynthesis. Because in CSIR NET paper from Unit 6, there are certain topics which are patented. In photosynthesis, most of the questions comes from the light reaction, from the dark reaction. Since both of these topics are very wide, so dark reactions we won't be covering today. Today we'll be covering just the light reaction, right? So in photosynthesis, we'll be covering light reaction today. So welcome you all. So at last, you can also download the free sample papers that would be provided to you from the link in the description. So the topics of discussion today is Unit 6, Photosynthesis, Light Reaction. So here we'll be discussing through revision tools such as Conceptica tables and Conceptica wheels, we'll be doing comparison between photosynthesis and cellular respiration. This is something which is very basic. Everyone should know, like, what is the difference between the photosynthesis and cellular respiration, where they are similar and where they are different from each other. Then we'll be covering the properties of the light used in the photosynthesis. This is also very important. What is the property of the light? Like, at which wavelength, which spectra of the light photosynthesis actually takes place. Then we'll be knowing the energy emission pathways. So there are various ways through which the pigments in the chlorophyll, they basically emit the light. So there are basically 4 ways. So we will be learning about all 4 ways. Then we'll be covering the chlorophyll. We'll be knowing the difference between the chlorophyll and the accessory pigments. What are their roles? Then we'll be doing the comparison between the absorption and the action spectrum. What does they actually signify? And then we'll be knowing about the light harvesting antennas and photochemical reaction centers. Then there will be stages of photosynthesis. There are 2 stages basically. There is the light reaction, and then there is a reaction where carbon dioxide is basically being fixed. It is a light independent reaction. Then we'll be doing the comparison between the cyclic and non-cyclic photophosphorylation, and we'll be knowing in which organisms these photophosphorylation actually takes place. Then is the comparison between the photosystem I and photosystem II. Then we'll be knowing the mechanism of electron transport. So this is the overview of the today's lecture and the discussion that we'll be doing. So firstly, there is the comparison between the photosynthesis and cellular respiration. So as we know that in photosynthesis, so as we know in photosynthesis, the reaction is basically this. Photosynthesis is an anabolic reaction where majorly the sugar is being formed from carbon dioxide and water, and oxygen is being released. Where when it comes to cellular respiration, it is actually a catabolic reaction where sugar molecule is basically being broken down in the presence of oxygen. It is an oxidation reaction where and after that carbon dioxide and water is being released with energy, right? And that energy is being utilized by us in the day-to-day activity. Now, 2 major phases of photosynthesis are the light reactions and the Calvin cycle, where basically the assimilation of the carbon dioxide basically takes place. That is the carbon fixation. And this basically occurs, photosynthesis occurs in chloroplast, while cellular respiration, it basically occurs in mitochondria and the cytoplasm, right? And the major stages of cellular respirations are glycolysis, Krebs cycle, and etc. That is electron transport chain takes place. Then it comes that the ATP is generated through the photophosphorylation in case of photosynthesis, while the ATP is being generated through oxidative phosphorylation in case of cellular respiration. Photophosphorylation means light is being used to generate ATP, and in case of cellular respiration, oxygen is basically being used to generate ATP, right? Then here what happens, there is a series of electron transport which takes place. The transfer of electrons takes place where when the photons they strike onto the chlorophyll pigments, then what happens, there is an excitation. These chlorophyll pigments, they excite and reach to a state where the electrons, when they go to the excited state, then they fall back to the ground state. At that particular point of time, some energy is being released, right? And in this, the end product in case of light reaction is basically NADPH. And in case of Calvin cycle, that is the light independent reaction, these products of the light reaction, they are being utilized in the Calvin cycle to produce the sugar moiety, right? And the process is anabolic, as I've already told. In case of cellular respiration, it is a catabolic process. It breaks down the glucose and uses the glycolysis and citric acid cycle to produce the NADH and FADH molecules, right? These are the molecules from which electrons are basically being released. They are the electron donors as electron carriers, right? Then comes the NADH and FADH called down to the ETC to power the ATP synthase enzyme. Now, these NADH and the FADH, which are the molecules which basically produce or provide the electron, these are the electron carriers. These are majorly being used for the synthesis of ATP in ETC reaction, that is electron transport chain, right? In case of cellular respiration, there are like around 36 to 38 ATP is being produced from one molecule of glucose or two pyruvate molecules, right? So these are the difference between the photosynthesis and the cellular respiration. Now comes the similarities. So both use some amount of ATP, right? ATP is being used here, and electron transport chain is present in both the cases, in plants as well as in animals. In animals, it basically takes place in mitochondria, and in plants, it basically takes place in chloroplasts, that too in the thylakoid membrane, right? Then comes the photophosphorylation. Yeah, phosphorylation basically takes place here in both the cases, and ATP synthesis takes place. And both of them require water to conduct these reactions. So this was the common differences and the common similarities between the cellular respiration and photosynthesis. I explained it with the help of the Venn diagram. So this is something which is very basic that everyone should know. Then coming to the next slide that tells about the properties of light used in photosynthesis. Now, before moving further, I would like to know where photosynthesis actually takes place, in which tissue. Like everybody knows that it takes place in the chloroplast, but chloroplast is an organelle. In which tissue of the photosynthetic plants the photosynthesis actually takes place? That's the question for all of you. Now, I would be inviting answers. The question is basically, what is the tissue in which the photosynthesis takes place in higher plants? Anybody? Yes, MT Noor Jahan. I'm asking about the tissue. Thylakoid is something which is present inside the chloroplast itself. It is a part of chloroplast, and chloroplast is an organelle. This is a tissue in which the chloroplast is present. Yes, Shivani, you are correct. That is the mesophyll cells. Inside the mesophyll cells, there are pigments present, which are known as the chlorophyll pigments. Inside that, only photosynthesis basically takes place. So active tissue in which the photosynthesis actually takes place in case of higher plants, these are the mesophyll cells, right? So end products, as we know, in case of photosynthesis are NADPH and ATP. These NADPH, these are the end products. Wait, let me change the color. So NADPH and ATP, these are the end products of the photosynthesis, right? And this NADPH, which is basically being produced in light reaction, this is utilized in the carbon CO2 fixation. In case of like where carbon assimilation basically takes place. We don't generally call it a dark reaction. Calling it dark reaction would be like something which is wrong, but calling it as light independent reaction would be much justified, because in standard books also, if you will see in T's and Ziegler for photosynthesis and for unit 6, I would refer you that you will be you study from T's and Ziegler. This is the best book for studying this particular unit 6, and in that also the 5th edition one. And in that, it is nowhere mentioned that the dark reaction actually takes place, because it's not actually the dark reaction. It is the reaction which takes place like without the presence of light. So calling it dark reaction would be wrong. Then, so this is the end product, and this NADH, which is being produced in the light reaction as an end product, it is being utilized in the carbon dioxide fixation in the light independent reaction, right? Then comes the properties of light used in photosynthesis. Now, as we know that wavelength, if you would have studied in physics, basically what happens, wavelength and frequency are two components which are majorly inversely proportional to each other, right? Now, as we know that for us, like for human beings, the visible spectrum in which the human actually can see things, it ranges from 380 nanometer to 740 nanometer, right? Now, the active photosynthesis, the spectrum at which the photosynthesis actually takes place, it lies from 400 to 700 nanometer visible spectrum. This is known as, right? So this is the active photosynthetic range. Where photosynthesis actually takes place. So this spectrum you need to remember. And coming to the equations which are utilized here, these are also the things which are very important. We have studied in our physics or 10 12 basically. So here it says that a photon has an energy which is denoted by capital E, is basically proportional to the frequency, right? So E is equal to h nu, where nu is basically a symbol for the frequency, right? Now h is the Planck's constant, and it is equal to the hc divided by the lambda. Lambda is the wavelength. So wavelength is the basically the it is a kind of measurement for the amount of light that is being absorbed or emitted. So the light that is being absorbed or emitted by the chlorophyll pigments, these are being measured in terms of the wavelengths, right? Now, another thing here is the relation of momentum p, small p, of the photon to its frequency and wavelength. So momentum is basically equal to the energy divided by c, that is the speed of light, equal to the h nu divided by c, and this is in turn is equal to the Planck's constant divided by the wavelength. So as we know that the shorter wavelength, wait a second. So short wavelength means that the frequency is high, right? Because as we know that the relationship between the wavelength and frequency is inversely proportional, right? So the shorter wavelength would have the high frequency, and in turn it will have the low energy, right? And these are basically the red light area, right? In the spectrum, the red light portion, the red light basically it has the shorter wavelength, and the frequency is high and the energy is low here. And in case of wait a second, yeah, it would be blue light basically. Sorry for the mistake. So it would be blue light. How blue light? As we can see from the equation itself here, that energy is directly proportional to the frequency and energy is inversely proportional to the wavelength. So from this equation, the shorter wavelength, the shorter wavelength would have the high amount of energy, right? Sorry, the shorter wavelength would have low amount of energy. Wait a second. Yeah, it's a short wavelength, so the energy would be high in this case. I'll write it again. So short wavelength means it would have high energy and it would have low frequency. Right? So look at it again. Now, as we know that the frequency is inversely proportional to wavelength, right? Since if we are choosing the shorter wavelength light, this short wavelength light will have high amount of, the frequency would be high, and in this case, the energy would also be high, right? Because energy and frequency have the direct relation, but this wavelength and the frequency have inverse relation. So this short wavelength is basically for the blue light, and opposite is for the red light, right? For red light, what happens is, red light has the longer wavelength and it has the shorter frequency, and thus it has the low energy. So this is for the red light. So did you all got this? Yeah, wavelength is inversely proportional to the energy and directly proportional to the frequency. Directly proportional to the frequency? No, wavelength is actually inversely proportional to the frequency, and it is in turn inversely proportional to the energy itself. Mahima? Yeah, make yourself right that wavelength is inversely proportional to the frequency, and this is in turn inversely proportional to the energy, right? E. Because here from the equation itself, you can see that E is equal to h nu, right? Nu is the frequency. So E is directly proportional to the frequency, while energy is inversely proportional to the wavelength. So shorter wavelength, those light which have the shorter wavelength, which will have the high amount of energy, and it basically lies in the like blue light is basically the one which has the shorter wavelength, and it has the energy is high in that case, right? And for the red light, it has the longer wavelength and the energy is low for the red light. So chlorophyll pigments, they basically absorb blue light. Then coming to the energy emission pathways. Now here we can see that if a pigment, it absorbs light energy, one of the three things will happen. Firstly, the energy is being dissipated as heat with no photon emission. So there are chlorophyll pigments, they have absorbed some amount of light. Now this, and they have reached from the ground state to the excited state. Now coming back to the ground state, what happens? They can dissipate the energy that they have absorbed in the form of heat, where there will be no photon emission. This is one way in which the energy emission takes place in case of plants. Secondly, what happens is there could be a fluorescence. So fluorescence is basically the energy is being emitted immediately as a longer wavelength, right? Here what happens that from the lowest excited state to the higher excited state, when electron is excited, when the light energy is being falling on it, then what happens is the energy emitted is being immediately emitted in form of longer wavelength. So wavelength would be high, which means that the energy would be low for that, right? And the chlorophyll's fluorescence is in the red region. So this is the red region basically. So in red region, what happens? The wavelength of the red region is very, like it has the longer wavelength. So, and the energy for the red region is basically low, right? Energy is low because both wavelength and energy has inverse relation. This is one of the way. So fluorescence is one of the way, then is the heat. Third way is the energy transfer. Now here what happens, the excited chlorophylls, they transfer its energy from one molecule to another. So there will be multiple complexes of chlorophyll molecules that would be present inside the chloroplast. Then what happens, that energy when moves from like in one chlorophyll molecule, when the energy falls, it transfers its energy to the another molecule. In this way, a chain of transferring of like electrons or the energy takes place. So this is another way. So this is the third way through which this energy emission takes place in case of plants. And fourth is the photochemistry. Photochemistry is basically energy here is being triggered by the chemical reaction. Right, whatever energy is being absorbed, it is being utilized in the chemical reaction where the glucose, that is the sugar molecule, is being broken down to produce the oxygen is being evolved here, right? Sorry, the glucose molecule is being produced from the carbon dioxide and water. So in this case, the photochemistry here majorly plays the role. So there are the four major ways through which the energy emission in plants here takes place. Now here is the energy level diagram is being given here. So here it is given that the absorption of the blue light. This absorption of the blue light, the energy is very high for the blue light, right? So here you can see from here till here, this absorption of blue light is taking place, right? And in case of red light, this have the shorter, like the energy is low and has the longer wavelength. So in case when from the ground state till the higher excited state, this is basically the blue region light, where basically the chlorophylls, they absorb the blue light. So reaching from ground state to the higher excited state, then again coming back to the low excited state. So first lower excited state is this one. This is the first lowest excited state from the higher excited state, where when the chlorophyll molecules, they emit energy, they emit it in the form of heat loss, right? Here the heat loss basically takes place and there is no photons which is being released. Now from this lowest excited state, at this particular higher excited state, the chlorophyll molecules, they are in a very unstable conditions, right? So therefore they need to come back to the lower excited state. This is the first lower excited state, where when coming to the lower excited state, the heat is being lost, the energy is being lost in the form of heat, and there is no photon which is being released. Now coming from lowest excited state to the ground state, which is the lowest energy state, here what happens, this is the absorption spectrum for the red light. This particular portion is known as the absorption spectrum for the red light. So here what happens, when the chlorophyll molecules, they moves from the lowest excited state to the ground state, then the emission of the energy, it basically takes place in the form of fluorescence, where there is no loss of energy by the emission of the light of longer wavelength, right? There is no energy loss, but in form of fluorescence, basically they release the energy, right? So here is the second figure, which basically tells us about the spectrum in which the blue light and red light basically occurs. So here it is being given that for the blue light, it is from 400 to 600 nanometer is the wavelength, and for red light, it is from 600 to 800 nanometer, that is the wavelength. So you can see that here red light is having the longer wavelength as compared to the blue light. So blue light have short wavelength, red light have longer wavelength, blue light having more energy, and red light having low energy, right? Now you would have been able to understand, right? Okay, Kumar, you're not getting it. Actually, we are here we are explaining about the photosynthesis spectrum, like energy emission, how it basically takes place. So through this graph, I'm explaining this. So here we have seen that the lambda, that is the wavelength, it is inversely proportional to the frequency. That is something which everyone knows, right? And this wavelength is also right. So what happens is, here it is being shown that the blue spectrum, it is having the blue light, it is having the wavelength from 400 to 600 nanometer, while red zone, it is having basically the wavelength from 600 to 800 nanometers. So here from the graph itself, we can see that the wavelength of the red light is higher as compared to the wavelength of blue light. So in turn, the blue light would be having the higher energy as compared to the red light. This much have you understood? And in case of red light, what happens when the chlorophyll molecules, when they move from the lowest excited state, that is the red light absorption spectrum, from here when they come to the ground state, then what happens? The energy, it is being released in the form of absorption, right? So energy is being released in the form of fluorescence. So this is the fluorescence energy spectrum which is being given here, and it is given that the red light is being absorbed in this particular wavelength. So this is something which you can infer from this particular diagram, this particular energy level diagram that is being shown here. And this is also given in your standard book, that is Tees and Ziegler. From there also you can go and like after this lecture, you can go and study for yourself, right? Then coming to the chlorophyll and accessory pigments. So there are two types of pigments which are present in case of plants. That is, the chlorophyll pigments are present, and then Okay, Kumar, Mukesh, I'll be going slow. So here I'm explaining that in photosynthesis, what happens in plants, there are two types of pigments which are present, which absorbs light. First is the chlorophyll pigment, and second is the accessory pigments. These have different roles. Now, all the green plants, the plants which like are involved in the photosynthesis, they have chlorophyll molecules present in them. And the range for the accessory pigments for the absorption of the light, it is around 400 to 500 nanometer, right? And this spectrum of light which is being absorbed, this wavelength of light which is being absorbed by the accessory pigments, it is not being absorbed by the chlorophyll pigments. So the light which is being falling, the 400 to 500 nanometer of light which falls on the accessory pigment, it is actually being transferred to the chlorophyll pigments first. So therefore, the name, it is being given to them as accessory pigments. So the diagram here shows, this is the chlorophyll molecule. So you can see here that this part, it basically resembles to the porphyrin ring that is majorly present in case of hemoglobin, as we have seen, right? So all the chlorophyll, whether it is chlorophyll B, chlorophyll A, chlorophyll C, all these chlorophylls, they have this kind of porphyrin ring that is present in them, which is majorly resembles to the porphyrin ring which is present in case of hemoglobin, right? In here, what happens? The magnesium is present at the central atom element here, which is present in the +2 state, while in case of hemoglobin, what happens? The central element is basically ferrous, that is in +3 state. This is the major difference. So, and the group which is different in case of chlorophyll, this is the diagram, this is the molecular diagram of the chlorophyll A, where the group that differs here is CH3 and C2H5 is present here in the porphyrin ring, while in case of chlorophyll B, CHO group is present, and while in case of bacteriochlorophyll A, that is the chlorophyll A also is being seen in certain kind of bacteria which conduct this photosynthesis reactions, such as green algae and all that bacteria. So what happens here, what the functional group which is different here is this particular group, right? So this is the major difference between chlorophyll A, the bacteriochlorophyll A, chlorophyll B, and the chlorophyll A molecule, right? Now coming to the accessory molecules. So accessory molecules here is the carotenoid. Carotenoids are the accessory molecules which absorb light in the range of 400 nanometer to 500 nanometer, right? These are having the structure is like, it is having a long chain, that is a straight long chain it is having with certain double bonds in between. You can see that. And these carotenoids, these are basically the integral part of the chlorophylls, which are majorly thylakoid membrane, which is majorly present both in the reaction center as well as in antennas. So about the reaction center and antennas, what is the difference between them, we'll be knowing later on. But major function of this carotenoid or this accessory pigment is that it transferred the light which falls on the chlorophyll, the light which falls, it absorbs the 400 to 500 nanometer of wavelength of light, and then it transfers to the chlorophyll pigments, right? So light is being transferred from these accessory pigments to the chlorophyll pigments. Light is directly, it doesn't fall on the chlorophyll pigments. Now here is the the light spectrum which is being given. Now, here it says the curve 1, it basically shows the spectrum of light for the absorption spectrum, basically for the bacteriochlorophyll A. Now, the curve 2, it says that it is for the chlorophyll A. So here you can see that the curve 2, it is for the chlorophyll A. This is basically for the bacteriochlorophyll A. So I'll denote it by BCHA, right? Third curve, it basically, you can see that the absorption curve is for the chlorophyll B. So absorption curve for chlorophyll B, you can see that is the highest. So it absorbs the highest highest absorption spectrum is for chlorophyll B, right? This is for chlorophyll B. And for fourth curve, it is for chloroerythrobilin, which is a protein which is present in case of cyanobacteria, right? And fifth is the bacteriokeratin. The fifth curve, it basically shows for the bacteriokeratin. So this one is the fifth curve for bacteriokeratin, beta keratin. So this is the spectrum for all these pigments that is being shown here. And yeah. So here is an experiment you can see, which was being conducted by the T.W. Eagleman. So he conducted an experiment where he measured the action spectra. So he took a Spirogara, right? A Spirogara organism he took, and what happened here? He used the spectrum of the light and projected on the spiral chloroplast. These Spirogara organisms, they also consist of the chloroplast, and in them the chloroplast is in spiral shape, right? So the spiral chloroplast of the filamentous green algae Spirogara. So what he did, he used the spectrum of light and projected on this. Then the observation that he had was that the oxygen-seeking bacteria introduced into the system. So oxygen-seeking bacteria were when he introduced the spectrum of light, which was being projected on the spiral chloroplast. After that, he introduced the oxygen-seeking bacteria, which are basically the aerobic bacteria, into the same system he introduced. And after that, what happened? Those aerobic bacteria, they were growing and they were collecting in bunch in the region of the spectrum where the chlorophyll pigments absorb. So wherever the chlorophyll pigments were absorbing the light, at that particular portion only, these aerobic bacteria, they were clustering. These are the aerobic bacteria, right? So it ranged from 400 to 500 nanometer, you can see, right? This is the wavelength of the light range in which the chlorophyll chlorophyll pigments, they absorb light. So here is the aerobic bacteria which were accumulating and growing. So the conclusion was that this action spectrum gave the first indication of the effectiveness of the light absorbed by the accessory pigments in the driving photosynthesis. So here it was being proved that the accessory pigments are the one who basically absorb light, right, in the range of 400 to 500 nanometer. And then they transfer it to chlorophyll molecules, chlorophyll A basically. And here only, wherever the absorption is taking place, the light is being absorbed, there the oxygen-seeking bacteria, they are growing, which means that the oxygen is evolving at that place. So there is a direct relation between the oxygen evolution and the absorption, right? So what we can infer from here is that the absorption region of the pigments, right, the chlorophyll pigments, is basically directly proportional to the oxygen evolution. So it was being seen wherever the absorption is taking place, the chlorophyll molecules, they are absorbing the light at 400 to 500 nanometer. There only, and at 600 to 700 nanometer, this is basically the absorption spectrum of the chlorophyll molecules. This is the absorption spectrum 400 to 500 for the accessory molecules, that is the carotenoids. So here basically the growth of these aerobic bacteria is taking place, which means that here this is the region where oxygen evolution is taking place, right? So in this way, the absorption spectrum is directly related to the oxygen evolution. So this much have you understood. How to clear this exam in first attempt. So like you have to be continuous with your preparation, and since like one exam you have you given the one that exam that happened on 17th? So like those people who have given their exam in 17th, if they are not giving themselves a break, and there are 2 more exams which are coming in this particular year, so in this very first like year you can clear the exam, since you're not giving yourself a break, and you have advantage that 2 more exams are coming for you, right? So Mukesh, 2 more exams are coming. The CSIR NET exams are also going to happen in this very year. So try clearing the exam in this particular year only. And in first attempt, you can clear the CSIR NET if you refer to the standard books. And the revision tools are also present here through which I'm explaining things. And now you can take help of some coaching institute. Then coming to the comparison between the absorption and the action spectrum. So this much have you, are you clear with it? This absorption spectrum and the comparison between the absorption spectrum and the oxygen evolution which is taking place. This experiment was basically being performed by the T.W. Engelman, where he used these the spiral chloroplast of the Spirogyra. Through this experiments, he majorly came to a conclusion where he said that the oxygen that is being evolved is directly proportional to the absorption region. Wherever the chlorophyll molecules they were absorbing the light, there only these aerobic bacteria they were growing, which means that they were basically releasing oxygen at that time. So in complete photosynthesis, at last what happens, the oxygen is being released. So that was the conclusion of this particular experiment, right? Then coming to the comparison between the absorption and the action spectrum. So what do you mean by the absorption spectrum and action spectrum? That we'll be studying here. So absorption spectrum is basically the amount of light energy that is being taken up or absorbed by a molecule as a function of the wavelength. Right. So in form of wavelength, right, light is being absorbed in form of energy only, but it is being measured in form of wavelength, right? So the molecule which are basically involved in absorbing light here are basically the chlorophyll molecules. Here it is talking about the chlorophyll molecules. So the light, yes. So absorption spectra is the amount of light energy that is being taken up or absorbed by these chlorophyll molecules as a function of wavelength. This is the first definition of the absorption spectrum, right? Then it says that the action spectrum is basically the magnitude of a response of a biological system to light. Like what it at which magnitude it will respond to the light that is being absorbed. Whatever light it absorbed, to which magnitude it will respond as a function of, this is also a function of wavelength, right? It is also being measured in terms of wavelength. So here it says for photosynthesis, it is constructed from measurements of oxygen evolution at different wavelengths, right? So the experiment that we studied in earlier slide, it was saying that wherever the chlorophyll molecules they absorb the light, there only the oxygen evolution was taking place and the bacteria were growing. From that, it was evident that the oxygen evolution is taking place at that particular area, right? Now it says that the action spectrum, the oxygen evolution is basically the way in which this action spectrum is being measured. Whatever amount of oxygen is being evolved, which is being measured in terms of wavelength, it tells the action spectrum of the reaction which is taking place, right? The magnitude of response which is taking place, that is photosynthesis, at which rate it is taking place. And second, it says that it identifies the chromophore, the pigments responsible for a particular light-induced phenomena. So this action spectrum, it basically tells that the chlorophyll molecules at which particular wavelength they are absorbing the light, right? So here in case of this particular diagram, it says that the absorbance and the oxygen evolution rate both are actually directly proportional to each other. So the graph for the absorption spectrum, you can say that it is coinciding with the action spectrum. The absorption spectrum and the action spectrum both are coinciding with each other. So action spectrum, from action spectrum, we can deduce that it basically tells about the oxygen evolution, and This absorption spectrum is basically tells us about the amount of light energy that is being absorbed by the chlorophyll molecules, or the molecules which majorly absorbs light. These molecules absorbing light in terms of wavelength, it is being measured, and this oxygen evolution, it is also being measured in terms of wavelength. So both of these graphs, they coincide with each other. So wherever there will be more amount of absorption, there will be more amount of oxygen which would be evolved, right? So this much have see. Okay, sorry. Now are you able to hear? Am I audible now? Okay, Amit Kumar, I'll be explaining this particular thing again. So what it is saying that majorly about the action spectrum and absorption spectrum, here it is telling the difference between them. So absorption spectrum is basically the amount of light energy that is being taken up or absorbed by the molecule as a function of wavelength. It is being written here. So absorption is basically the molecules, that the chlorophyll molecules in case of photosynthesis, these are the molecules which absorb a particular wavelength of light, right? And after that absorption only, they like the chlorophyll molecules, they go to the excited state, then they come back to the ground state, and energy is being released in different forms, as we have studied, in form of heat, in form of fluorescence, and that energy transfer which is taking place. So all these things takes place, and by this only the chemical reaction takes place, and the anabolic reaction where glucose is being produced from the carbon dioxide and water takes place. And in this only, along with the production of the sugar, there is the oxygen evolution which takes place, right? So oxygen evolution is basically measured in terms of action spectrum. So this graph basically tells where it is written that the action spectrum, this action spectrum basically tells the amount of oxygen that is being evolved, right? This is the oxygen evolution. This is indicating the oxygen that is being evolved when the chlorophyll molecules, they absorb light. So more the absorption of spectrum for the chlorophyll molecules, more will be the oxygen that would be evolved. Oxygen evolution basically takes place, it is the end thing which takes place in case of photosynthesis, right? So action spectrum tells about the oxygen evolved, while absorption spectrum is basically tells the wavelength of light the pigments basically absorb, right? So here it says that for photosynthesis, it is constructed from measurement of oxygen evolution. And it is also being measured in terms of the wavelength, and it identifies the chromophore, that is the pigments that are involved in light absorption, responsible for the particular light induced phenomena, right? So at what particular wavelength chlorophyll A molecule it absorbs light, or at what particular wavelength of light chlorophyll B it absorbs light, or those accessory molecules it absorbs light, that is being indicated by this action spectra, and also tells the amount of oxygen that is being evolved during this process. So this much have you clear? Okay, Anupam, he says it's clear. Then we'll move moving further. So this is a quick reminder for all of you that shorter batch, it would be starting from 4th March, right? So in this batch, we'll be providing with you a lot of facilities would be there. There will be RAID classes by CSIR NET experts, and interactive online classes would be there. 24/7, there would be doubt solving, and hard copies would be provided. Study materials would be provided in form of the concise notes, which you can use for revision and for clearing your concepts. And video backups will be there. So if you want to know more about the Shaurya batch, which is starting from the 4th March, you can contact us in the toll-free number that is flashing on your screen, or you can mail us, right? So the team will guide you how to proceed. Now here comes the light harvesting antennas and the photochemical reaction centers. So these are the two important things which are present in case of chloroplast. So here what happens, the diagram, it says that the pigment molecules are present in the form of antenna complex. So all the pigment molecules, they arrange themselves and are present in the form of complex here. These are known as the antenna complexes, and these pigment molecules, they absorb light of a particular wavelength. And energy that they are, like the light that they absorb and the energy that is being emitted, they transfer that energy to the reaction center, right? Reaction center are basically of two types, the PS1 and PS2, of which we'll be studying. like in upcoming slides. So what happens, this reaction center, when the energy is being transferred here, there would be a donor electron, there would be a donor molecule which would be present, and it would donate an electron through the reaction center to the acceptor molecule, right? So here it says that the many pigments together serve as the antenna molecules, right? So pigments, all the chlorophyll pigments that are present in case of chloroplast, what happens, they together, they form an antenna complex inside the chloroplast, inside the thylakoid membrane, and then the antenna complexes, they collect light. So light basically falls on this antenna complex, and they collect the light and they transfer its energy to the reaction center, right? Then what happens in this reaction center, the chemical reactions basically takes place. So here is the chemical reactions which takes place in the reaction centers, right? To store the sum of the energy, like some amount of the energy which is being like transferred to these reaction center from these pigments, these antenna complexes. Some of it is being lost in the form of heat, but most of it is being transferred to the reaction centers, where they basically, what they do, they form, like they produce a chemical reaction in which this energy is being like it is being stored in the stored form. The energy is being stored in case of a reaction center, and by this transferring the electron from the chlorophyll pigments to an electron donor. Now this transfer, what happens after this transfer takes place, this gives an energy to the electron donors. This electron donors, what they do, they transfer the electron from the electron donor to the electron acceptor. So electrons, they like go from the lower excited state to the higher excited state, right? And electron donor then reduces the chlorophyll again. Then what happens, this chlorophyll molecule, it is being reduced by the electron donor again, right? So what happens, since this donor molecule is donating the electron, it will be in its oxidation state, it will get oxidized, and the acceptor molecule, which is basically being accepting the electron, it will get reduced, right? So that is what it is saying, that the electron donor then reduces the chlorophyll molecule, that is the acceptor one. So this much have you understood? So Mukesh will be discussing the questions here only, and you can contact this toll-free number now that was given earlier. In that toll-free number, you can contact, and every help would be provided to you, right? So this much have you understood, the light harvesting antennas and photochemical reaction centers, the difference between them. So then coming to the funneling of the excitation from the antenna system toward the reaction center, right? How does basically this energy is being transferred, how the excitation is majorly taking place from the antenna complexes and the reaction center. So about antenna complexes and reaction center, we have studied like what are these and how the like light is being transferred from the antenna complexes to the reaction center, and then the electron donor, it transfers the electron from to the electron acceptor, right? Reducing the electron acceptor. Now it is saying the excitation, the energy gradient, how the energy gradient is being formed here. Now it says that the first figure, it says that the excited state energy of the pigments increases with the distance from the reaction center, which means that those pigments which are present near to the these reaction center, it will have the low amount of energy, right? And those reactions, those pigments, those antenna pigments which are present farther from the reaction center, those will be having the high amount of energy. How? Because these carotenoids are the one who would be the first like absorb, they will absorb the light first, and then they will transfer to the chlorophyll molecules, as I have told earlier, and they absorb the wavelength of light that they absorb is basically ranges from 400 to 500 nanometer, right? Now what happens, the light will directly fall to these carotenoids, which are the accessory pigments. These accessory pigments, like obviously they will be having the high amount of energy since the light is directly falling to them. Then what happens, these accessory pigments, they transfer the energy to the chlorophyll molecules. First chlorophyll B and then chlorophyll A. So chlorophyll B would be having like yeah, so firstly the carotenoids would be having the largest amount of energy. The absorbed light is high for them, and the energy would also be high. For after that comes the chlorophyll B, and after that comes the chlorophyll A. So with the distance from the reaction center, it can be measured that which chlorophyll will have the highest amount of energy, or it has absorbed the high amount of light, right? So first comes the carotenoids, then come the chlorophyll B, then comes the chlorophyll A. Then it says that due to the energy gradient excitation transfer toward the reaction center, its energy is favorable. So here what is happening is that the energy gradient, the excitation is basically, the transferring is basically takes place from the complex to the reaction center. So this is, the energy is lowering down from high to low. So this is the highest point and this is the lowest point. So this energy is basically favorable for the transferring of the energy from the pigments to the reaction center. That's what it is saying, that due to the energy gradient, because of this energy gradient, the excitation, wait a second, yeah, the excitation transfer toward the reaction center is energetically favorable. So this type of reaction is energetically favorable, right? Then comes the B part. So where here it says that some energy is being lost as heat to the environment by this process. Now, when the energy is being transferred from this these antenna complexes, these pigments, to the reaction center, some amount of this energy is being lost in the form of heat, right? No photon is being released; it is being lost in the form of heat. And rest everything, the energy is being transferred to the reaction center. So in optimal condition, what happens, almost all the excitations absorbed in the antenna complex, it can be delivered to the reaction center, right? So this is what it is saying here. So when the light falls on the carotenoids, it gets to the excited state. From this excited state, it gets to the lower, like it transfers its energy to the chlorophyll B. So here basically energy transferring is taking place, right? One of the way that I told, emission of energy takes place. One is the energy transfer also here. So here pigment molecules that transfer energy from like one molecule to other. So this is how it is taking place. Firstly, the carotenoids are the one which absorb light of 400 to 500 nanometer. Then they transfer their energy to chlorophyll B. Then they transfer to chlorophyll A, and then at last to the P680, that's the reaction center, right? So here it is written that the energy is being lost as heat during the excitation transfer. So during this transfer, basically the heat loss take place. The heat is being lost, and after that, what happens when from the chlorophyll A, the energy is being transferred to the reaction center. The energy of the reaction center excited state available for storage. So whatever energy is being transferred from chlorophyll A to this reaction center P680. This is being this energy is being stored for the chemical reaction to take place, right? For the carbon dioxide and water to convert into the sugar molecule. That is the chemical reaction for which the energy is being required, and that is being stored in P680. This reaction center, it is being stored, right? So this much have you understood? Now, coming to the stages of photosynthesis. So there are two stages of photosynthesis. One is the light phase, and another is, since it is written dark phase, actually it's not a dark phase, right? It is basically the light independent reaction here basically takes place. You cannot call it the dark phase. Nowhere in the standard books also it is not written that it is basically the dark phase. So correct it, okay? So one is the light phase, another is the light independent reaction, light independent phase. So what is the major difference between both of these stages? It is being given here. So firstly, we'll be watching the overview of this particular photosynthesis reaction, right? So light is falling here and the photophosphorylation takes place. Water is being split and oxygen is being evolved, and the ATP that is being generated and NADPH that is being generated here, it is being utilized in the light independent reaction, right? So this is the light independent reaction where basically the Kelvin cycle plays a role, and here basically the carbon dioxide fixation takes place, right? So what happens? Photophosphorylation is the light reaction. This is basically the light dependent reaction, and in photophosphorylation, after splitting of water molecule and oxygen is being evolved basically here. So what happens here, the end products of the light reaction which is being produced, that is the ATP and NADPH which is being produced, it is being utilized in the light independent reaction, where Kelvin cycle basically takes place, where oxygen fixation takes place and the formation of the sugar molecule takes place here, right? So these are the two stages. So photophosphorylation is the light reaction and the Kelvin cycle is basically the light independent reaction, right? This is the overview, and coming to the comparison between the light phase and the light independent phase. So light phase, it basically, or light reaction basically occurs in grana, and light independent reaction, it occurs in the stroma, right? Grana is called the appressed or the stacked region. It is basically known as the appressed or the stacked region. So granas are basically present. If we draw the structure of chloroplast is there. So chloroplast molecule is there. So granas would be present here, right? These are known as the granas. So in granas, basically a light reaction takes place, and these are present in the form of appressed or stacked region, right? Stroma is also known as the non-appressed or the unstacked region. This is basically the unstacked region, stroma region, which is present inside the thylakoid. And here basically the light independent reaction takes place, that is the Kelvin cycle takes place, and here only the carbon assimilation takes place and the sugar is being formed, sugar molecule is being formed. Then coming to another difference, where it says that light reaction, it is also known as the light dependent phase, and the dark reaction is also known as the light independent phase. Actually, it is known as the light independent phase only. Then comes the light, about the light phase, it says that it does require the semi-permeable membrane and occurs only across a semi-permeable membrane. So light reaction doesn't require semi-permeable membrane, while the light independent reaction basically occurs, or it requires the semi-permeable membrane to take place, right? And the products of the light reaction, this is the major thing here. This is the whole crux of both of these comparisons. In light reaction, the products that is being formed is ATP and NADPH, and oxygen is being evolved here, as we have seen in this case, this overview, which I have shown. And here only what happens, these products, these ATP and NADPH, they are utilized as a in case of this light independent reaction for the carbon dioxide assimilation and production of glucose molecule, that is the sugar moiety, right? So here it says that the photosynthesis have two stage. It is a two-stage process. One is the light dependent reaction takes place, and another is the light independent reaction, where the carbon fixation takes place. So in light dependent reaction, it requires the direct energy of the light to make energy carrier molecules, right? So here directly the light falls on it and it forms the energy carrier molecules. Energy carrier molecules here basically the ATP and NADPH it is talking about, right? This is the energy carrier molecules, NADPH and ATP. So these energy carrier molecules which is being produced in the light reaction, they are being utilized in case of light independent reaction. The product of the light reaction are being utilized, and here these light molecules, they are being used for to form the carbon-carbon covalent bonds of the carbohydrates, right? So the carbohydrate moiety, that is the glucose which is being produced, in that the carbon-carbon covalent bond, for the formation of that bond, this energy carrier molecules are being required, which are the product of the light reaction, right? This much, are you clear with it? So Mukesh, you can go, the video would be there in the YouTube only. You can go back and check it again if you're not clear with the earlier slide, right? Coming to the cyclic photophosphorylation and non-cyclic photophosphorylation, comparison between both of these, right? So photophosphorylation is basically the light reaction which takes place in case of organisms. So there are two types of photophosphorylation light reaction takes place. That's first is the cyclic photophosphorylation, and second is the non-cyclic photophosphorylation. So there are different organisms in which these cyclic and photophosphorylation, non-cyclic photophosphorylation takes place, right? So non-cyclic photophosphorylation majorly takes place in autotrophs or the higher plants, while cyclic photophosphorylation, it takes place organisms such as cyanobacteria, purple sulfur bacteria, or green sulfur bacteria, right? All these prokaryotes are the one in which the cyclic photophosphorylation takes place. Now, yeah. So first, it says that the cyclic photophosphorylation is also called the N-oxygenic form of photosynthesis, as it does not involve any oxygen. So here no oxygen evolution takes place, right? In case of cyclic photophosphorylation. In case of non-cyclic photophosphorylation, this is also known as the Z-scheme pathway. So here the oxygen evolution takes place, and it is also known as the Z-scheme pathway, because the pathway or the events which takes place and the way in which the electrons is being transferred, it looks like a Z-scheme, right? Then it says that it involves only the photosystem 1. In case of cyclic photophosphorylation, only photosystem 1 is there, right? And in case of non-cyclic photosynthesis, both photosystem 1 and photosystem 2 are there, required. Then it says in cyclic photophosphorylation, only product of cyclic photophosphorylation is ATP. So only ATP is being produced here, while in case of non-cyclic photophosphorylation, 3 products are important here. 3 products are being formed. First is the ATP is being produced, another is the NADPH is being produced. Both of these are the energy carrying molecules, and oxygen is being evolved, right? Then comes the electron donor. Here is H2S. So sulfur H2S is basically the electron donor here. From sulfur molecule, basically the electron is being transferred, right? And in case of these products contribute to carbon dioxide fixation in the light independent reaction, right? The products which is being formed here, the energy carrier molecules ATP and NADPH, which is being produced in non-cyclic photophosphorylation, it is being used as a product. It is being used in case of the carbon dioxide fixation in case of Calvin cycle, right? Which is a part of light independent reaction. Then comes that it says that occurs only in the lower organic cyanobacteria, as I have told already, that lower organisms only this cyclic photophosphorylation takes place, while non-cyclic photophosphorylation, it basically takes place in autotrophs, in higher plants. Then it says, this diagram basically says tells us about how cyclic photophosphorylation actually takes place, right? So it is a schematic diagram of cyclic photophosphorylation. So it says that it is found in the eukaryotes and some primitive photosynthetic bacteria, right, like cyanobacteria. So here what happens, there is the antenna complex is here, right? So as we know that the light directly falls on the antenna complex first. That is firstly the accessory molecules, then the chlorophyll pigments, right? This way the energy transfer takes place in the antenna complex. So here majorly what happens, the photosystem 1 is only involved here. Both the photosystem 1 and 2, they are required in case of Z scheme, that is non-cyclic photophosphorylation. So photosystem 1, it absorbs light of which wavelength basically? Can anyone tell me, like photosystem 1, it absorbs which wavelength of light? Beta bioscience. He says that the bonds, does bond formation releases energy? No, bond formation, it requires energy actually. This carbon-carbon bond which is being formed in case of like carbohydrate during the carbohydrate formation, at that particular point of time, the energy is being required, and this energy is being provided by the product of the light reactions, the energy carriers, that is the ATP and NADPH. Yes, Aishwarya, it's right. So photosystem 1, it absorbs the wavelength of light which it absorbs is basically of 700 nanometer, right? So this is the wavelength of light which photosystem 1 absorbs, right? Energy from the 2 photons is coming here. Now, this energy, this light which is being absorbed, this energy is from the antenna complex, it is being transferred to the primary acceptor, right? So there will be 2 electrons which are being transferred here. This primary acceptor, it will transfer this electrons to in the form, the energy is basically being transferred in the form of electrons, right? So this will transfer the electron to the ferredoxin, then plastoquinone, then chromophore complex, cytochrome complex would be there, where basically what happens, the ADP is being converted into ATP, right? So its final product is ATP. So here the product, only one product is being formed, only one energy product is being formed, that is the ATP, right? And only one photosystem is involved, photosystem 1, which absorbs which wavelength of light? 700 nanometer. So this is the reaction which takes place. After what happens from this chromophore complex, then it reaches to the plastoquinone and then to the reaction center again. So this is a whole cycle in which the electron transfer, the energy transfer basically takes place, right? So from what happened, light came, light fall upon the antenna complex. Antenna complex is basically present in the photosystem. It is talking about the photosystem 1 here right now. So 700 nanometer is the wavelength of light which is being absorbed by the photosystem 1, which is and these photosystems are basically the reaction centers, right? So what happens from these antenna complexes? The energy is being transferred in the form of electrons. First is the primary acceptor. Primary acceptor here basically would be this reaction center it is talking about, right? From this reaction center, the electron would be transferred, the energy would be transferred in the form of electrons to the ferredoxin. Ferredoxin is an electron acceptor. Then it donates to plastoquinin, the electrons. 2 electrons are basically being transferred here. From plastoquinin, it transfers to the cytochrome complex. And this cytochrome complex is the place where basically the ADP is being converted into ATP, right? So only one energy carrier molecule is being produced in case of cyclic photophosphorylation, that is ATP. And after this, what happens? The energy is being dropped down and comes to the lower excitation state, and it is being transferred to the plastoquinin. And from plastoquinin, it again, like in a cyclic form, it travels to the reaction center. So here it is saying this is the reaction center. This yellow colored molecule, it is saying that this is the reaction center. So and from antenna molecules, what happens? The energy is being transferred from one pigment to the other. Then it is being transferred to the reaction center, then to the primary acceptor, and then likewise it happens, right? So this much have you understood? Frat, Sunny Sharma, yes, of course. Frat also will be teaching, like in like we'll be conducting classes for frat also, right? Don't worry. Your command is being noted. So then coming to the non-cyclic photophosphorylation. Yes, in this non-cyclic photophosphorylation, as we have studied, it will basically takes place in higher autotrophic plants or higher plants in which the photosynthesis takes place, right? Then what happens is the non-cyclic photophosphorylation, the electrons is being transferred in the form of the Z scheme here. So firstly, there is an oxygen evolving complex is present here. So firstly, what happens? The energy falls on the antenna complex. Water is being split here. So sunlight is the only energy through which the water can be split. No other energy in the world can split water, right? So oxygen evolution here taking place after the water is being splitted. So oxygen evolving complex is there. Right, then what happens? This energy which is being produced due to this like splitting of water, then what happens? It is being transferred to the P680. So after the P680 is the photosystem 2. So photosystem 2 is basically it absorbs light at a wavelength of 680 nanometer, right? So light falls on this reaction center. Then this the electrons are being transferred, the energy is being transferred in the form of electrons. So firstly, this photosystem 2, it will get excited. From the ground state, it will reach the excited state. After here, it will be transferring the electrons from it will be donating the electrons to photon. Then it will be transferring the electron to the QA molecule, then QB molecule. Then there will be a cytochrome B6F complex to which it will be transferring it. So this order you need to remember, because in exams actually what happens, sometimes the question is being asked in the form of like what would be the order of the transfer of electrons which takes place in case of Z scheme, right? Then what happens from this cytochrome B6F complex, the energy is being transferred to the plastoquinin. From plastoquinin, the energy is being transferred to the photosystem 1. This is the another reaction center which absorbs light of 700 nanometer, right? So light also falls on it. 700 nanometer light falls on it. It absorbs light, and apart from that, it is all the electron transfer. The energy transfer is also being taking place from the P680 till this P700. That is the photosystem 2. This photosystem 2 reaction center, it gets excited and gets to its excited state. Right. This asterisk symbol basically shows the excited state. Then what happens? Then it transfers the electrons to A0, A1 molecule, FeSx. Then FeSa, FeSb, ferredoxin. Then what happens is, after that, the last molecule, the product which is being formed through this non-cyclic photophosphorylation, that is Z scheme, is the NADP plus gets converted into NADPH. This is the energy molecule which is basically being required in case of the other light independent reaction, right? So this is the whole Z scheme which is taking place. The order you need to remember. PC is plasto. Yeah. Yes, Sridevi. The PC is basically the plastocyanin, right? Then what happens? There are certain inhibitors which you need to remember, right? So DCMU is one of the inhibitors of this non-cyclic photophosphorylation. So this DCMU basically acts upon where it basically acts upon the QA/QB part. Here basically DCMU inhibitor takes place, like inhibits the non-cyclic photophosphorylation electron transfer. So DCMU acts here in between QA and QB molecule. So what happens is here you can see that the QA is the electron donor and QB is the electron acceptor here, right? So QB is getting redoxed and this is getting reduced and the QA is getting oxidized. So here in between these this process, this energy transfer, this electron transfer only, the DCMU acts, right? And another is the paraquat. This is another inhibitor which basically acts in between FeSa and FeSp. So here it basically acts, and both of these inhibitors are very important. Questions related to inhibitors are also being asked, like at which particular step these inhibitors majorly act upon, right? So what happens when paraquat inhibits this at this particular step, then what happens? No NADPH is being produced. The energy carrier molecules are being not produced by when both these inhibitors acts upon this non-cyclic photophosphorylation. So inhibiting it leads to the inhibition of the production of the NADPH molecule, right? Then coming to the differences between the two photosystems, photosystem 1 and photosystem 2. These are the reaction centers which are present both in non-cyclic, these both are required in non-cyclic photophosphorylation. So photosystem 1, it says that the components of the photosystem 1 reaction center are organized around two major core proteins. So two major core proteins are present here, PSA and PSAB, right? Around these core proteins only, these components of a photosystem 1 is being present. Second, for the photosystem 2, it says that so photosystem 1, it basically absorbs light at 700 nanometer, and this photosystem 2 680. This thing everyone should know, right? The structure of the photosystem 2 comprises of the two parts of electron transfer domain and water oxidation domain. So there are two basic major parts here present in case of photosystem 2 that absorbs light of 680 nanometer. One part basically consists of the electron transfer domain, from where the electron transfer basically takes place, and another is the water oxidation domain, so where the splitting of water basically takes place. After water splitting, oxygen is being released, right? So two major centers are present in case of photosystem 2. Then comes the minor proteins, the PSA-C and PSA-N are also present. So these are some kind of proteins which are present, components of the photosystem 1. So these are present in photosystem 1, and in photosystem 2, it says that it has a special structure called the OEC. OEC complex is basically, or the oxygen evolving or the water oxidation complex is present here, right? So that's what I have said, the two domains are present. One is the water oxidation domain, another is the electron transfer domain. So second, the water oxidation domain is basically known as the OEC complex, that is the oxygen evolving complex, right? So that undergoes photolysis of water to give oxygen and the 4 protons. So 4 H plus molecules are being released here, right? Evolution of oxygen takes place. 1 molecule of oxygen is being evolved, and 4 molecules of proton, that is H plus molecules, is being evolved here, right? After the photolysis of water takes place. Then coming to the electrons are transferred from the plastocyanin. So here the electron is being transferred from plastocyanin to this photosystem 1, that is P700, and then to the chlorophyll molecule A0 and to the plastoquinone molecules A1 to ferrous center, Fe center, and finally to the iron sulfur protein, that is the ferredoxin. So this is the order they have given. So firstly, what happens here, it says that the electron is being transferred from plastoquinone and it is being transferred to the P700, that is photosystem 1. After that, it is being transferred to the chlorophyll molecule, that is A0, and then to the plastoquinone A1, right? And after that, it is being transferred to the Fe center would be there, and finally to the iron sulfur protein would be there, Ft, that is ferredoxin complex. Here it is being transferred, and from here basically what happens, the NADPH P plus is being converted into NADPH, right? This is the energy carrier molecule which is being produced here. And after this, it says that the PS1 is a part of both cyclic and non-cyclic photophosphorylation, right? So what we have seen in cyclic photophosphorylation, only one photosystem is present, that is the photosystem 1, and photosystem 1 is also present in case of non-cyclic photophosphorylation. So it is present in both kind of photophosphorylation reaction, right? Photosystem 2, for it says that the core protein are D1. Here the core protein are D1, D2, and peripheral protein are the CP43, CP47, CP26, and 29. The number represents basically the molecular weight in Daltons. So here the numbers, it basically represents the molecular weight in Daltons of the protein that is involved here. So the core proteins or the components of photosystem 1, it is being given here, 2 is being given here, right? And it says that the photosystem 2 is a part of only non-cyclic photophosphorylation, right? Photosystem 2 is not present in case of cyclic photophosphorylation. This much is clear, right? Then comes the distribution of the protein complexes, right? How the protein complexes are majorly being distributed in thylakoid membrane. So it is being explained here. So here it says that you can see that LHC2 trimer is basically here. These molecules are present here, and the major things which you need to remember here is the distribution of PS2, PS1, and ATP synthase, right? So for PS2, you can see that it is evenly distributed in the thylakoid membrane, right? And for PS1, what you can see is so here it says that PS2 is located predominantly in the stacked regions of the thylakoid membrane. So PS2 is majorly present in the stacked region. These are the stacked region, right? These regions are the stacked regions. These are the unstacked regions. And these are the stacked regions. Means these are present in between the membranes, right? So these are known as the stacked regions. So PS2, this one, it is saying that green color one, this PS2 is located predominantly in the stacked regions of the thylakoid membrane. So it is evenly distributed in the stacked region. While PS1 and ATP synthase, they are found in the unstacked region. So here you can see that ATP synthase is there, right? This unstacked region. And here only that PS1 would be present here. So PS1 and ATP synthase, these are majorly present in the unstacked region of the thylakoid membrane. Then comes the cytochrome B6F complexes are evenly distributed. And here we can see that cytochrome B6, this F complex, it is evenly distributed throughout. It would be present both in stacked and unstacked region. So this is the basic overview of how the distribution of the protein complexes majorly is taking place in thylakoid membrane, right? Then this is the mechanism of electron transport, how this electron transport reaction is majorly being taking place inside the thylakoid membrane, right? So here you can see that this is the structure of the chloroplast. So inside the grana, there would be thylakoid membrane where basically this reaction, this electron transport reaction, photophosphorylation reaction basically takes place, right? So here it says that the light strikes the PS2. Firstly, the PS2 is the reaction center where the light strikes. And this PS2 is having the two domains, right? One is the electron transport domain and another is where the water splitting basically takes place, right? And light strikes the PS2, causing the transfer of excited electron to the primary electron donor, right? So what happens? The transfer of electron takes place and the electrons are being transferred from the PS2. So firstly, when the light strikes on the PS2, it gets excited. It moves from lower excited state to the higher excited state, right? These electrons are being replaced by the splitting of water. So water, when it splits, it produces 4 H plus ions, no? So these electrons are being replaced by these when the water splits, and which releases O2 molecules. So O2 molecules are being released here. Oxygen molecule is being released when the water is being splitted, and O2 is being released as a waste product. So here the first is being taking place here. So this is the first one which it is talking about. Another, it says the second step, it says the excited electron, it travels down an electron transport chain, right? The electrons which are being excited state, the electrons are being produced when the photolysis of water is taking place and the light is being falling on the PS2. At that particular point of time, the electrons which are being excited, it travels down the electron transport chain which occurs, right? So this process pumps H plus ions across the membrane into the thylakoid. So what happens when the electron transport is taking place? The H plus ions, they are being crossing this plasma membrane from the stroma to the lumen. So inside the lumen, the concentration of H plus ion would be high, and outside, like in the stromal region, the concentration of H plus ion, it would be low, right? So it happens along with the electron transport. So light excited electron is in the PS1, right? In PS1, it says the light excited electrons, when the light falls in PS1, the light is falling on PS1, the light excited electrons, what happens? These electrons are being replaced by those coming from the electron transport chain. So the electrons which are coming from the electron transport chain, it is being replaced, these replaces the electrons which are produced due to the falling of light on, striking of light on PS1, right? Then it says that the backflow of the H plus ions out of the thylakoid powers the ATP production. So what happens? Here the electron transport is taking place, and the H plus ions are coming from this stroma to the lumen, and inside the lumen, the H plus ion is the concentration of H plus ion is very high. And what happens when the ATP is being produced, like after the production of the energy carriers, ATP is also the second product which is being produced here. So the hydrogen ions which are present in high concentration in case of lumen, these hydrogen ions are being transported through this ATP synthase enzyme, right? F0 F1 complex it has. So from this ATP synthase complex, the hydrogen atoms or hydrogen ions, they are being transferred, and as a result, the ATP is being produced. The ADP is being converted into ATP, right? So this is the whole reaction which is taking place. Two products are being formed, NADP and ATP, after this whole photophosphorylation reaction. This is how it basically takes place in the thylakoid membrane. And these two products which are being produced in this photophosphorylation light-dependent reaction, these are being utilized in the carbon fixation reaction in case of light-independent reaction, right? So this is the whole mechanism that I have told you. Then comes the regulation of the photon capture and the photo damage repair. So what happens is, with this much of intensity of light, when the light is falling on the chlorophyll molecules, it leads to the damage of the molecules there. So first line of defense that they use to prevent themselves from this photon damage is basically, firstly, the suppression of the damage by quenching the axis of excitation as heat. So heat is being released, right? In antenna complex, when the light is being falling, it can lead to the damaging of those antenna complexes. For that, what happens? Some amount of energy that is being captured from the light, it is being released in the form of heat, right? The second line of defense, if this first line of defense doesn't work, then what happens? The second line of defense comes, where scavenging systems, they eliminate the reactive photoproducts. So reactive photoproducts are basically the redox reaction takes place in reduced products which are being formed. That is such as peroxides, and all these free radicals are being produced. These are being eliminated, right? Thirdly, what happens? The photo products can damage if this second line of defense also fails. Then the photo products which are being produced, these the reactive products which are being produced, that is the hydrogen peroxides and all these free radical molecules which are being produced inside, they can damage the D1 protein. So D1 protein, as we have studied when we are comparing between PS2 and PS1, we have studied this is a core protein in case of PS2. This protein gets damaged, right? The damaging, what does it do? It leads to the photo inhibition. The photophosphorylation process, it inhibits, right, due to this damage. Then what happens? The D1 protein is excised. If this damage happens, then they have their repair mechanism. How do they do the repairing these of chlorophyll molecules? What happens? The D1 molecule protein which is being damaged, it is being excised out from the PS2 reaction center, and it is being degraded, right? And another D1 molecule is being synthesized. It is being resynthesized, and it is being inserted in the PS2 reaction center to form a functional unit, because plants require photosynthesis to take place, and photophosphorylation is the first major stage which takes place for the production of the glucose molecule, right? And if photophosphorylation is not taking place, then light independent reaction will also not take place. So to prevent and for themselves from this damage, they basically do this reparation mechanism that they apply is being given here. These are the steps basically involved. So in the form of diagram also I have explained it. You can go through this diagram in your standard book. And coming to the questions. So these are the 2 questions that I have picked up from the CSIR NET paper related to the particular topic that we have discussed till now, related to light reaction, right? So this is a kind of homework for all of you. So next, I would be coming on Wednesday. On Wednesday, we'll be discussing, and Wednesday I'll be coming up with Unit 8, and there we'll be discussing these particular questions from Unit 6, right? So these are the 2 questions which are kind of homework for all of you. So thank you very much. And now at last, you can also download the free sample papers, and the link is being given in the description. First, there will be a feedback form that you have to fill up. After that, you can download your free sample papers, right? So thank you very much, and all the best to all of you.